we're good. So, before we go on to just looking at random amino acid sequences and which ones might be useful antigenically, as far as for developing the pathogenesis of the SARS coronavirus, I want to track back just a little bit to what I was doing earlier. So as I said earlier, I aligned a lot of different spike proteins from different species of coronavirus, and I also aligned it up with the multiple versions of the POMC protein, which is the protein that generates a lot of the peptides that are important for they're important hormone peptides, and one of them, ACTH, is known to be attacked by your own antibodies as a result of a influenza infection or a coronavirus infection. So I reasoned that it'd be extremely useful to do this alignment like this. That way, if there are any changes going on between species, if those changes are conserved in some way, then you can look at those semi-conservations and then be able to compare them with the semi-conservations of the other. Now, I did this, and then I also did it without the other coronavirus spikes. And one of the things that I really want to mention with this is if we are just focusing our attention on the SARS-CoV-2 coronavirus spike and see how it might be able to interact, or should I say cross-react, with, or create antibodies that cross-react with it and certain peptide hormones in the human body. Well, my concern right now is there is one sequence that pops out when you just look at SARS, but if you look at all of the other spikes, there is spikes from different coronaviruses. You don't see any of that alignment in this area. But if you just focus on the SARS coronavirus, you do see that alignment. And so here we have, this is the NPP peptide, and it keeps on going down here to Sirac, and I don't know exactly where it ends. I can actually get that up for later. But I want you all to be aware is this is the very beginning of the spike protein of SARS coronavirus 2, the N-terminal side. And here, this sequence of the beginning portion of the spike protein, this is actually very uncannily, this very uncannily matches the NPP peptide of the human body. So going back to the POMC, which is the big protein that produces a lot of peptides in it, if we go down to the different sections, and I might have skipped it already. 1 second. Here we go. So between residues 27 and 102, this is the NPP peptide. And so here we're starting pretty much at 27. This is 27. This is a part of that particular peptide, and this is the N-terminal region of that peptide. Now, what does this particular peptide do? Well, this peptide is an important part of the adrenal cortex. So it's an adrenal growth factor. So the adrenal glands are endocrine glands that produce a variety of hormones, including adrenaline and the steroid aldosterone and cortisol. They are found above the kidneys, and each gland has an outer cortex which produces steroid hormones and an inner medulla. Regardless, this is a naturally occurring endogenous metabolite, and it is a precursor to these other peptide hormones. So actually, I'm going to do a quick Google search to see if oh, okay, so these are the melanocyte stimulating hormone pieces. Okay, that's good to know. So as you can see, yeah, as I said earlier, biochemists love their acronyms, and that makes it very difficult sometimes to keep track of everything. So here you can see the NPP, and it's this large thing. And then one of the sections here they say is melanotropin gamma. So it's one of the hormones that help what's it called, stimulate the melanocytes. If I am understanding this correctly. And so apparently there's a bunch of different peptides even within this NPP that are associated with the production of melanin, which I don't know if y'all heard of the two Chinese doctors who started they started producing an extreme amount of melanin. Well, I don't know if it was melanin, but basically their skin turned black while they were in comas. I mean, this might be something that could explain why that ended up happening. Regardless, so this is going back to it. Okay, so this is actually I would have to look. I'm going to go look up to see if this is actually a highly conserved sequence in a lot of different creatures, if that makes sense. Because so obviously, as a hormone, it's highly conserved, but I'm curious to know if it's conserved as a completely different structural component of different proteins that have absolutely nothing to do with being a hormone. I mean, that it could be that this is just a very stable and good sequence for a particular shape, and different enzymes and different proteins will use that shape. But yeah, to me, this is not something that should be taken lightly. When you have an alignment of 1, 2, 3, 4, 5, 6, 7, 8, 9, so 9 amino acids that are exactly the same, somewhat spread out, and then you have 1, 2, 3 amino acids that have very similar chemical properties as well. And then, yeah, I think that this is definitely going to be physiologically relevant. So I'm going to look into it a little bit, and hopefully, I shouldn't say hopefully, but maybe there is an effect there. And to me, that's actually almost a little scary to see, because remember, there are 20 proteogenic amino acids, right? So, I mean, it's not a exactly random process whenever you have these amino acid sequences. Uh, but approximately, if you were to take any two amino acids between two proteins, if you're going to line it up, and granted, this is lined up through an algorithm, so take it with a grain of salt. But if one of them matches, that's just a 1 in 20 chance. And so you can easily slide around on an amino acid sequence, and then you'll easily run up across one of them. But to find two, that's a 1 in 20 to the second power chance. Or 1 to the 400th. Yeah, it would be 1 to the 400th. And if you have three that are identical, it would be 1 to the 2 to the third power. And if you have 9 of them that are exactly in the same spot, it'd be 1 to 20 to the 9th power. So that is extremely unlikely, unless it was an extremely conserved shape. I mean, yeah. So sometimes you can have quite a few exact residues, and yes, they're unlikely, but since you're able to sweep across the amino acid sequence, eventually you can find hits that are very similar because you're just given a lot of options. But if you have as many as this, then it's no longer a matter of, it'll no longer be a matter of having enough amino acid sequences, because you would need so many amino acids. You would need a string that was so incredibly long that it would, be more DNA than what the Earth has to provide. So if it's not coming up from chance, then it has to come up from some conservation of some sort. Now, granted, evolution can cause these things to just appear, but yeah, I'm gonna have to really mull over this for a little bit, because it is possible that a virus can infect a creature and then be able to steal some of its genetic material, especially since, so if there was another RNA molecule floating around that encoded for this particular section of the amino acid sequence, then it's possible that there could be some sort of crossover event, or some way that you can get the RNA-dependent RNA polymerase to accidentally take it up and incorporate it. But yeah, I think at least with this particular sequence, where it's leucine, serine, 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 or leucine, and then serine, serine, and then Q, glutamate. Wait, let's go look at the proteogenic amino acids. I always have to double check, because I believe Q is glutamate, but this is stuff that you learn and then you forget, and then you look it up again, and then oh, glutamine, so not glutamate. Glutamate is E. So going back, so but a valine instead of the glutamate, and then asparagine and LTT. Yeah, so leucine, threonine, threonine. Yeah, I think that this is something that we should consider, especially since it's so early in the amino acid sequence. So it'd be very likely, especially during degradation of your spike protein, it would probably be one of the things that will be cleaved and then just be free floating as a free peptide during the processing. Ugh. Yeah, I just wasted a lot of time on that. But yeah, I want y'all to know that. And then also, just so you are all aware as well, down here where we have more sequence alignments, I want to also say that this cleavage site of the human POMC is aligns pretty well with this cleavage site of the S sub 1 or S sub 1 subunit and the S sub 2 subunit of the spike protein. So this is frequently where the spike protein will be cleaved by protease, which will help it enter the cell, or sometimes it can be cleaved even before it has attached to the cell as a post-translational modification of the virus proteins before it's even fully developed. So there's that, and then also there was another right about, I think it was here, this KR over here. So this is where the this is the S sub 2 prime cleavage site, which this is just another cleavage site on the actual spike protein, and it, it tends to be a little those lysines tend to be conserved, except right here it's shifted over a little bit to be aligned differently from what I remember from this one. So keep in mind, it is algorithms trying to come up with all this stuff, and so it's very difficult. Okay, so here we go. So this KR, this is an additional cleavage site, and so it's somewhat polybasic, right? You have two lysines, and then you have an arginine, so that could be easily cleaved. And then it aligns up with the lysine down here. Oh, but that's the actual cleavage site. We're just learning a lot right now. This is why we have computers look at all this stuff, because if you keep on looking at this, you're going to be doing this for freaking ever. Anyways, continuing on, but what I want to do right now is just go over how you can get inferences of what might be important amino acid sequences from this blast. And then if you really want to check, you can go to pepBank, and this can tell you if the peptide that you are looking at has a physiologically important role in the body, or at least in whatever host organism you're talking about, or whatever organism you're talking about. So we end up, a lot of our research that we do, we will put the peptides onto a database describing what these peptides do. And a lot of times we do peptide screening. So if there's something that we want to discover, a lot of times we will look up many, many, many different types of peptides on this particular, for a particular system. And if we get any hits, or if we have any peptides that influence the activity of the system that we're looking at, then we will say, okay, these peptides are doing something to the system, and then that will get uploaded onto a database. Or oftentimes, we will also take peptides, and then we'll just change a few of the amino acids on them, and then we can determine which amino acids have more importance than others on a peptide in reference to changing the system in question. So here, I'll give an example, but this is a really long and tedious process. So, but here we'll do the SSQC. We'll see if there's anything, SSQC, if there's anything that aligns at all with that. So here we have SSQC, LVK. So it doesn't quite fit or align here, but we'll just do this for our own edification. So here we have activation of, okay, so this is the name of the peptide. This is the full sequence of what this peptide is. And going down here, this is an abstract where someone had done some experiment on this particular peptide. And so here it's activation of protein kinase C, or CAMP-dependent protein kinase, increases phosphorylation of the C-ERBA encode thyroid hormone receptor and the V-ERBA encoded protein. So I'm going to try to understand this in the few minutes that I can. So the C-ERB proto-oncogene encodes a nuclear receptor thyroid hormone, T3, which is believed to stimulate transcription from specific target promoters upon binding to cis-acting DNA sequence elements. The V-ERBA oncogene of avian erythroblast virus, or AEV, encodes a ligand-independent version of this nuclear receptor. Oh, okay. The verb A product inhibits terminal differentiation of avian erythroblasts, presumably by affecting the transcription of specific genes. We show here that the two distinct sites. Can't see the whole thing. Okay. We show here that the C-ERBA encoded nuclear receptor is phosphorylated on serine residues of two distinct sites. One of these sites defined by the limit tryptic phosphopeptide. This is retained on the verb A encoded P75 GAG VE or VA. Oh my god. Biochemist. This site is located in the amino terminal domain of these molecules. 21 amino acids upstream of the DNA binding region. Phosphorylation of the site in both P46C ERBA and P75 GAGV ERBA is enhanced tenfold following the treatment of cells with activators of either protein kinase C or CMP-dependent protein kinases. Since CAMP-dependent protein kinase okay. You know, I cannot follow this at all. So this is something that I'm gonna have to look up the actual paper and then really digest it. But yeah, I can't. So what this sounds like or looks like to me is that this peptide, so this is a part of the thyroid hormone, the NPP, which is the thyroid hormone, and it sounds like that this amino acid sequence might be produced by avian viruses, which can mimic this in some way, shape, or form. So do not quote me on this. I'm going to have to read that paper myself. But this is, yeah, this is how you would theoretically find pieces of the amino acid sequence that might be physiologically relevant. Now, this is the long, tedious way to do it. But luckily for us, pepBank has made a much better method of doing this process. So I'm going to X that out, and I'm going to get the peptide database out. So here I will do this just so that we are aware. So what I did here was I took the sequence of the SARS-CoV-2 spike protein. And so here, this is a sequence, and I did an alignment, and this tells me that, yeah, maybe there are important amino acid sequences that are used to do some form of mimicry of host organism peptides. Okay, so go here, you can put in this amino acid sequence, and each one of these letters is another amino acid, and then you can change what types of algorithms you're going to use to discover these amino acids. So, well, let's play around with this. I'm going to do PAM70. I have no idea what this matrix is or what this algorithm is, and but we're just going to go with it. So search. Oh wow. Okay, so and what happens is you are given a list of all the amino acid sequences that have at least a little bit of homology. And holy crap, this is a lot longer than the other one. So I used a different algorithm for this peptide database, and it wasn't as long. So I will go through. Actually, I'll go through this new one with you because I might learn something during this entire process. So here they will give you the score, which is some semi-quantitative way of telling you how similar the proteins are, and then there's an E value, and you know, frankly, I don't know all the math in this, so we are just going to jump into it. But the E value, the smaller the E value is, the much more likely, I believe it's there's more homology between the sequences, and then the score might be a function of all that. I don't know. So here, And here's a good one. So here would be an example of, so this is a sequence that is 100% the same. And so one would be like, oh, that's important. But if you go to the PubMed, or if you go to the paper that describes the sequence, you'll find that your internet is actually really bad. Okay, so continuing, that the highly conserved domain within a severe acute respiratory syndrome coronavirus spike protein S2 domain with characteristics of a viral fusion peptide. So it makes sense that this is conserved because this was a peptide that was described in the original SARS, and we know that it is useful, or I shouldn't say we know that it's useful, but it has characteristics of a viral fusion peptide. So it has characteristics of being important for fusion, and we know that it's conserved in other coronaviruses. So that makes sense that when we take the SARS-CoV-2 protein, we get a perfect alignment with this other SARS fusion peptide. But that doesn't say much, right? Because it's important that we have the tool to be able to find that. But you need to look further down. You can't just have 100% be what you're looking for. You want to look for things that are between 50 and 90%, because then those things aren't exactly the same, but they might be doing similar functions. So either they could be mimicking the function, or they could be trying to antagonize the function. There's really a lot that can be done by being similar to a different sequence, but not exactly the same. So, this is especially true if sequences have multiple functions that they have to do. So, a sequence in one virus or in one creature, maybe there's only a few amino acids in that sequence that are important for its function, but maybe it can change the other amino acid sequences so that those amino acid sequences can fulfill a different function. So that would be one way that you could conserve some amino acid residues while changing the others so that you can do more with less amino acids, with less, what would the word be, nutrients, with less resources. So, rambling. So I realized that I'm going to be doing all of this anyway, so I might as well record it for posterity. And to share my thought process on all of this. So, continuing on, let's go down to this section. So this is taking it from amino acid 209 to 218, YIK, WPW, YIWL. So, just to verify, I'm going to go back to not this one, not that one. We'll go to this one because I like this one the most. We're going to take this and we are going to search. And as you can see, we have this sequence. It doesn't have anything to do with the other alignments that we did, but maybe it's showing something that we're not looking for. So here, we'll click. This will take us to the paper that had characterized this sequence. So peptides derived from HIV-1, HIV-2, Ebola virus, SARS coronavirus, and coronavirus 229E exhibit high affinity binding to the formal peptide receptor. So here they're saying that there is a peptide, and let me make sure that they're describing the peptide. Yeah, so here we have peptides that show up in different viruses throughout the throughout the virus kingdom. And so here we have the formal peptide receptor, which let's do a quick Google search on that. I searched this up before, but The formal peptide receptor, or FPR, belongs to a class of G protein-coupled receptors involved in chemotaxis. In humans, there are 3 formal peptide receptor isoforms, each encoded by a separate gene, that are named FPR1, 2, and 3. These receptors are originally identified by their ability to bind N-formal peptides, such as N-formal methionine, produced by the degradation of either bacterial or host cells. Hence, formal peptide receptors are involved in mediating immune cell response to infection. So maybe these are on the white blood cells themselves to help tell them where to go. Let me just double check this. These receptors may also act to suppress the immune response under certain conditions. The close phylogenetic relations of signaling and chemotaxis and olfaction was recently proved by detection formal peptide receptor-like proteins as a distinct family of fomonasal organ? Okay, I've never heard of that before. Chemosensors in mice. FPR is now properly accepted as FPR1 by the International Union of Basic and Clinical Pharmacology. Let's look at the function real quick. As well as positively. But what does it do? So keep in mind, this is a receptor that goes through the cellular membrane, so it's not free floating, it's stuck to the cellular membrane. So what it sounds like, to a degree, I can't think this fast. What it sounds like is that this peptide interacts strongly with this particular receptor, and this receptor is important in having cells move. And then it also has something to do with the sense of smell. So maybe that if we're going to step back and take a wild assumption, and we'd actually have to scour the literature to see if this is actually a link, maybe the reason why some people have issues smelling and tasting, for instance, is because of this particular peptide that has high homology in the SARS-2 coronavirus and is conserved in a lot of other viruses that are also pretty darn serious. So maybe this particular peptide, if it ends up getting released in some way, shape, or form, then theoretically it might be interfering with your ability to smell. You know, that I'm making a large assumption right now, and I would normally really look into it, but I think we should continue. But just keep in mind, this is sort of my thought process of like, how do we go about trying to figure out what this beast is? So X that out, X that out. Continuing on. So here, another sequence, and this is in the 773 region. So let's see if it was relevant to yeah, so this is within the section that is associated with, at least to a degree, the peptides that were a part of the POMC that are in the region of the subunit 1, or S sub 1 and the S sub 2 subunit boundaries. So as you can see, this is the RRAR, this separates subunit 1 from subunit 2, and this is the subunit 2 region, and it's close to the subunit 2 prime region, except it's not on it exactly, because I think that is a little bit further here. So let's go to this and see what it has to teach us. A fluorogenic peptide containing the processing site of human SARS coronavirus S protein, kinetic evaluation, and NMR structure elucidation. So, it's going to be a prime target for SARS regimen. So they're saying that besides furin, that there are other proteases that are able to do cleavage in these areas. So this is something that really needs to be considered as well. If these protein sequences in this area are also susceptible to proteolytic cleavage, then it could be a technique of the virus to take advantage of your proteases to release these peptides into the environment. So let's just read a little bit longer. Involved in this cleavage event. This data suggests that the cleavable peptide bond is located within the most exposed domain. This is supported by the nearby turn structure, several strong weak NMR-ROSI correlations, which is just a type of NMR study. Yeah, so I think they're just saying that this part seems to be exposed to the environment, and that might lead it to being easily cleavable. So this isn't telling us much about this peptide itself. It's just saying that the sequence on the SARS coronavirus is at least somewhat exposed to a degree. So this could be important as far as producing antibodies, right? So if these areas are exposed to the extracellular environment, this can tell you that it might be important for antibody production. But then another thing we need to consider is if we're making antibodies against the spike protein in these areas, they are going to align with, they might cross-react with the peptides, the peptide hormones that your body is going to be producing as well. So that didn't say much, and keep in mind we're still very high in the percentage rate, and as we go further down, hopefully we get a little bit more variety into what the peptides of different organisms. So X that out. So here we have, so this looks similar to this. Ooh, no, this looks similar to one we saw earlier. So yeah, this looks similar to this one, so hopefully it is different. Oh, nope, it's the same paper. So it's the same paper that described two peptides that were very close together. So, oh, here is one, the hepcidin precursor, or the liver express antimicrobial peptide. So here, this is something that I did read another, I think it was a ChemArchive paper. It wasn't exactly, it wasn't peer reviewed yet, or it was in the process of peer review, or they just wanted out there to circumvent peer review, because sometimes the peer review process can be excruciatingly slow. And the peer review process happens when you release these papers out. Then people read them and they say, no, this is complete bullcrap, or this is actually something that we need to consider, and you can completely circumvent the official peer review process. But here we have the hepcidin precursor. So they're saying that, or here, the identity is pretty darn close with part of hepcidin. And hepcidin is a small peptide, and actually I'll just look it up for you so that you can see it for yourself. So hepcidin in your body works on iron similarly to how, what would the word be? Insulin, similarly to how insulin works for blood glucose. So when hepcidin gets released into the body, if your body produces hepcidin, that is telling your body to sequester the iron. So, or So let's just, I don't think I really need to go too much into detail with that. So I'll go through the clinical significance. There are many diseases where failure to adequately absorb iron contribute to iron deficiency and iron deficiency anemia. The treatment will depend on the hepcidin levels that are present, as oral treatment will be unlikely to be effective if hepcidin is blocking enteral absorption, in which case per- parenteral iron treatment would be appropriate. What is parenteral? Oh, iron salts and iron pills. Okay, but isn't that also oral? Okay. Would be appropriate. Studies have found that measuring hepcidin would be a benefit to establish optimal treatment. And although it is not widely available, C-reactive protein is used as a surrogate marker. So, yeah, just know that hepcidin is extremely important in iron metabolism and making sure that your iron levels are kept in check. So, um, I did read a paper where someone was worried that because this area, so this is the CC, I'll just look up CCSG. So find P. So here, this near the very end of the spike protein, so in the C terminus of the spike protein, we have a lot of cysteines. So cysteines are amino acids that are used to create covalent bonds with other cysteines. So it is a way you have your amino acid chain. If you want to uh, really solidify your amino acid, your structure of your protein or the structure of your peptide, using cysteines that have these covalent, really strong bonds is very useful to accomplish this. So this is, but I guess I shouldn't read too much into it because these cysteine-rich amino acid sequences are found throughout nature very frequently, um, especially as toxins. So I guess I'm not really not being very, what would the word be, optimistic with this. So, but just know or be aware that if this area of the amino acid sequence is mimicking hepcidin to a degree or at all, it could be convincing the body that the hepcidin levels are high. Therefore, we should start pumping iron into the cells, which I personally think would be very smart for a virus to do because if a virus requires a lot of iron cofactors, or if the to so a lot of iron can be used for the chemical processes of the enzymes in the cell, and then also iron might be able to competitively inhibit other ions that might not necessarily be good. So for the virus, so for instance, zinc ions we know do a pretty good job at inhibiting the RNA-dependent RNA polymerase of the SARS-2 coronavirus in vitro, but it's very difficult to get zinc into the cells of a living organism because the cytoplasm or the inside of the cell tightly regulates what metal ions get in and out of it, if possible. So when it comes to um, so if you are, when it comes to what would a strategy that a virus could be employing by getting a bunch of iron into its system, well, iron could be replacing zinc, or it could be being uh, made in such a high or concentrated into such a high concentration that even if you do have zinc within the cell, that the iron is going to push the zinc out of the way. So that would be a possible strategy. And then, another strategy is down the road after the virus has already duplicated and multiplied as much as it wants. Maybe it thinks maybe if you start having antibodies that are attacking this sequence, well, then they're going to start attacking the hepcidin in your body as well. That would be an example of how it works. Or the sequence might block hepcidin's ability to function by fitting into the receptors that hepcidin usually fits into. And so it just always blocks hepcidin activity. It's really hard to predict what is going to happen from an amino acid sequence, but because it could either positively or negatively affect or do nothing at all, except it can tell you that maybe it does something, and maybe you should devote your attention to this particular thing. So moving on, I'm going to skip these because these are just the ASPDs. They're not actual papers. They're different types of peptide studies. So genome, polylead protein. So yeah, this has no paper, so I won't be able to delve into it at all. This has no paper. Here it's looking like these sequences are part of the capsid proteins of different viruses, and so there seems to be, some homology between what one virus does and what a different virus does as well. But granted, this doesn't look that similar to me, but it could be. So moving down. Oh, so here, this is the mu conotoxin, PNIVA. So you might have heard of cone snails. Cone snails produce small proteins that absolutely wreck the system. And these small proteins are also very cysteine-rich areas that, so basically the cysteines are forming a lot of the knots within this peptide, and then it becomes a very stable molecule. And there's a lot of variety within these molecules to be able to do a bunch of different functions. So here, they're saying that this end part of our spike protein looks very similar to a conotoxin, but that's not really saying too much because a lot of things look like conotoxin. Hell, the hepcidin that we were talking about earlier that deals with iron metabolism, that also looks like a conotoxin to a degree. It has a bunch of cysteines in it that are involved in making a very tight structure. So moving on, let's go to this one. What is this? It doesn't have any. Okay, so this is just another conotoxin one. Oh. So precise epitope mapping and characterization is important for the development of a subunit vaccine to identify epitopes in the principal neutralizing determinant or PND within the V3 loop of human or HIV. Sera were screened in a direct ELISA assay with a coding peptide consisting of this. So here they are doing epitope mapping, or they're looking for peptide sequences on a virus that are really good at interacting with antibodies and interacting with the immune system and creating a very strong response. So here, this would indicate to me, unfortunately, that this sequence does in fact look a little bit similar to a sequence loop on HIV. Granted, I mean, you shouldn't say, oh man, you have HIV in your system, or HIV in the virus protein. This is probably a sequence that shows up a lot in a lot of different viruses. So for instance, I believe Gaga is the, yeah. So for instance, this GAGAA sequence is not just in the SARS-CoV-2 coronavirus, it is also in a bunch of other coronaviruses as well. So, oops, did not mean to do that. So what that can tell you is that this sequence is important and it shows up in a lot of different viruses. So here, MLCCM, that looks important. Let's look on this. Let's do the similarity and positive. No, not that one. MLCCM, okay, well. Okay, so here, MLCCM, not something that lined up earlier, but we'll go with it. An amino terminal domain of the growth-associated protein GAP43 mediates its effects on filipodal formation and cell spreading. So filipodal formation, so these are like little feet that come out of cells to help them move around, if I remember correctly. So an amino terminal domain of the growth-associated protein GAP4 mediates its effects on filipodal formation and cell spreading. So they're saying that there is a peptide in this GAP43 protein that is important for mediating the effects of the entire protein itself. So I'm not going to go through the whole thing, but LCC, MRR, if that is the MLCCM, MLCC, okay, so yeah. So we find that the amino terminus MLCCM is an important contributor to these effects on cell shape. So, I mean, one could, I don't know if it's a neuronal peptide that is important for neuronal growth and nerve terminal plasticity. It is enriched on the inner surface of the growth cone membranes, localization that may depend upon polymetallization of the cis3 and cis4. It is a major substrate for protein kinase C. So this is an important neurological peptide. Now, what I'm interested now is this protein secreted? And I'm sure if I read the paper, it would tell me, but I'll do a quick Google search to see this protein. Is it something that is produced within the cell, or is it something that is secreted out? Because if it's secreted out, that could tell me that it is subject to antibodies, or more easily subject to antibodies. So load please. So it looks like it's made in the RNA. Yeah, so a lot of it is made within the brain. So it doesn't look to me as though it is a secreted protein. So that doesn't mean that, oh yeah, it's a nervous tissue specific cytoplasmic protein that can be attached to the membrane via dual palmatization sequence on the cysteine 3 and 4. So this palmatization, this was oh good, it brings up a photo. So here you can see this is palmitoyl coenzyme A. It's basically a very long fat, and then there is a way to, well, it's a fatty acids, but instead of the acid group, it's connected to CoAs. This is able to transfer this long fatty tail onto cysteines of different proteins. And if you do that, then this long fatty group is going to actually move the protein into the cell membrane, or it'll move it into a membrane, because there are different membranes within the cell. So the sequence targets this to lipid rafts, and yeah, this is an example of what a lipid raft is. So, and it's considered to play a key role in neurite formation. So it sounds like this is not exposed to the outside world, or the out of cell world. So what this tells you, not much. You just have a very short sequence. here. And granted, I guess we could look up to see if the neuronal growth, you know what? That would be something to do right now. In fact, actually, did I already do that? I, I did, actually. Okay. So let me make sure I have the right protein. No, I think this is a completely different protein. So what was the sequence? MLCCM? Yeah, so this is a different protein. So yeah, it doesn't line up. So, I mean, this didn't really tell you much, but you have to go through a lot of these things to find the diamonds in the rough, right? So this is another one. Let's see if it didn't mean much for that. Maybe it means something for, nope, still the same thing. MLCCM. So here's one issue is that a lot of the peptides described have been described because they were in one paper has done a bunch of different permutations of an amino acid peptide sequence, trying to figure out which parts are important or not. Excuse me. So you can get multiple papers for the same amino acid sequence. So here, 88%, and it's kind of long. So let's see if this is an important one. Oh, yeah, yeah. This might, I remember reading this, this might have something to do with influencing thrombin. So as we all know, the SARS coronavirus has CoV-2. One of the complications of it is a blood clotting disorder. Now, I do believe a lot of that blood clotting has to do with insufficient iron metabolism during the time, but viruses often employ multiple techniques to get the same job done. Because if one technique ends up not working, it still has a few others up its sleeve. So here, alpha thrombin-induced human platelet activation results solely from the formation of a specific enzyme substrate complex. So let's take this sequence. So LQPR. So LQPR. So LQPR. So it's in this part of the amino acid sequence. It's pretty early up. So not much to say there. But prior studies using the mutant thrombin or thrombin quick 1 indicate that this protease induces a maximum platelet aggregation and intraplatelet calcium 2 plus fluxes as agonist concentrations where dissociable. Equilibrium platelet binding is undetectable and led to the conclusion that thrombin interaction with its platelet receptor is best described kinetically by formation of an enzyme substrate complex. This conclusion was substantiated further by the present studies by demonstrating that both thrombin quick 1 and thrombin mimicked the thrombin receptor agonist peptide in the induction of the platelet activation dependent events required for functional prothrombinase assembly, and that a rabbit antibody raised against this particular sequence. So was this the sequence? It's hard to LDPR SFL. Yeah, so this is the sequence. So what they did was they injected this peptide into a rabbit, and that rabbit ended up starting to produce antibodies against this peptide. And this pentadecapeptide represents amino acids 32 to 46 in the platelet thrombin receptor substrate, receptor/substrate, and spans the thrombin cleavage site. So this is a peptide that is a part of the substrate, which I believe is fibrinogen, but maybe it's not. I'm not exactly sure. I'll have to look at a better visualization to help get this straightened out in my head. But this is an important amino acid sequence that does get cleaved by thrombin, it sounds like. What particular peptide it is, it might not even be on fibrinogen. So it might be a completely different protein that thrombin is also able to cleave. So, I mean, here we have PR and then a serine, so that probably is where the cleavage is happening, but don't quote me on that. So it inhibited both thrombin and thrombin quick 1 induced platelet activation responses equivalently. The antipeptide antibody had a more pronounced inhibitory effect on the rate of the thrombin-induced response rather than the magnitude of the response, suggesting competition for the cleavage site, consistent with the observation that pretreatment of metabolically inhibited platelets with thrombin, which was removed by washing, eliminated specific antibody binding due to removal and/or masking of antibody epitopes. Concentrations of the antipeptide antibody that inhibited thrombin and thrombin quick 1 induced increases in intracellular calcium flux by as much as 97% did not alter the dissociable equilibrium binding of this. So these combined data indicate that the hydrolytic event initiated by thrombin or thrombin quick 1 interactions with the platelet receptor substrate for the thrombin is unrelated to the dissociable equilibrium binding of thrombin to the membrane sites described previously by classical receptor ligand binding analysis. So this is pretty difficult for me to like fully grasp. So what it sounds like is that there is a substrate. Let's look up prioritizing the mutant thrombin quick 1 indicate that you know, I'm just gonna leave this. Just know that whatever this effect is, whatever is happening to thrombin and the thrombin quick factor, this sequence that was used to create antibodies against a certain portion on either the receptor or something, just know that this is similar enough that it might actually be doing something. So, wait, was this the PubMed? No, it was this one. So yeah, this sequence that is found on SARS spike protein is, you know, somewhat similar or pretty darn similar to this. So perhaps it is going to be causing a similar effect. Continuing down, let's go here. An hour and 2. So mechanisms of arginine proproglyphy inhibition of thrombin. So this is another thing. So why is it talking about that amino acid sequence when this is the amino acid sequence in question? Mechanisms of arginine, proline, glycine, phenylalanine inhibition of thrombin. So let's go through. Investigations determine the mechanisms by which this sequence inhibits thrombin-induced platelet aggregation. So this is an amino acid sequence that inhibits thrombin-induced platelet aggregation. So what I'm going to do is just make sure. Yeah, so I don't have that sequence in the SARS database or in the SARS spike protein. Inhibits thrombin-induced platelet activation, and high concentrations of this inhibit thrombin-induced coagulant activity. So high concentrations of this inhibit thrombin-induced coagulant activity. So if you have this, it inhibits coagulation. Just get that in your head. A lot of positives and negatives to worry about. So high concentrations of this inhibit that, and this binds to the active site of thrombin by forming a parallel beta strand with this. So this peptide ends up forming a beta strand, and just know that a beta strand is a secondary structure of protein. So it's a way to orchestrate the amino acids. And as I said earlier, it wasn't this video, but there are multiple secondary structures. One of the secondary structures is a helix, where the amino acids are in a helix. Another one is when they're in a strand, and when they cycle back and forth and make a strand. So here they're saying that this amino acid sequence will form a strand, and then this strand will interact with another strand that is in thrombin. So parallel beta sheets with this and interacts with histine, aspartate, and all of these. So this competitively inhibits alpha thrombin from hydrolyzing the SAR-pro-ARG paranitroanilide with this constant. So I believe this is a what would the word be? Fluorescent molecule, and this is a amino acid sequence that thrombin is very good at recognizing. So the proline arginine, PR, I believe that might be the preferred cleavage site of thrombin. Continuing, other mechanisms were sought to explain why this inhibits the thrombin activation of platelets at concentrations below that which inhibits its active site. So soluble, this blocks biotinylated this of the thrombin cleavage site on protease activated receptor, or PAR1, from binding to the peptide this. So the soluble recombinant extracellular domain of PAR1 blocks biotinylated this binding to this, but this, oh god, I need a drink. And related forms prevent the thrombin-like enzyme thrombocytin from proteolizing this at concentrations that do not block thrombin's active site. These studies indicate that this is a bifunctional inhibitor of thrombin. So, what I believe they're saying, it's pretty difficult, is this is pretty good at combining with thrombin. This is a completely different peptide, not one that you find in SARS. This is a good peptide at blocking thrombin, but it is also good at binding with this of the thrombin cleavage site on the protease activated receptor. So this is also really good at binding with this peptide sequence, which is on the protease activated receptor, or PAR1. Okay, let's follow this. I think I'm starting to get that. So somehow those this will interact with this and then block the thrombin cleavage. I see. Now, with all chemicals that are inhibiting enzymes, there is or I should say all chemicals at all. If you have any interaction, sometimes the chemicals will be interacting and sometimes they'll just be floating around freely. And there's a certain concentration of, if you keep one constant, you can increase the concentration of the other. And there's a concentration at which most of these ends up being bound by one of these. And what they're saying is, at very low concentrations of this, you were still, at this RPPGF, you were still able to do a lot of inhibition of the thrombin activity. So here you have the thrombin and you have the RPPGFs, and a lot of times you use the RPPGFs to inhibit the thrombin by binding with it directly. But they said that at lower concentrations than necessary, or than what you would expect, it was still blocking a lot of thrombin activity. So what could cause that? Well, if it's binding with if it's not just binding with thrombin, but it's binding with the substrate of thrombin. Oh, okay, okay. I think I'm starting to get this now. If it's binding with the substrate of thrombin, and it might actually bind even better to the substrate, then that would be another way to do. That'd be another way to inhibit the thrombin from working correctly. Now, I don't know why. I don't know why that it or where they discovered this peptide in the first place. So we'll do a quick Google search and stable metabolic fragment of Bradykinen. So this is a peptide that is produced from the metabolic fragment of Bradykinen, which I believe is part of your body. It's a potent endothelium. Oh, this is interesting. Okay, so it's a potent endothelium-dependent vasodilator and mild diuretic, which may cause lowering of the blood pressure. It also causes contraction of non-vascular smooth muscle in the bronchitis and gut, and increases vascular permeability, and is also involved in the mechanism of pain. So going back, this peptide, which inhibits thrombin, and then also will bind to the substrate of thrombin, that receptor, that PAR1. This is an active peptide that floats around in your body, and it's basically a waste product, if you can call it that. It's a waste product of the bradykinin, which is a potent endothelium-dependent vasodilator. So, and that binds with this sequence pretty well, okay? And this sequence is mimicked to a degree. I mean, we don't know if it's actually a function of the SARS coronavirus spike at all, but that peptide is somewhat emulated by a section of the spike protein. Fascinating. So if you could imagine, if you have this going on, floating around in solution, or maybe it could be on the full spike protein itself, then this sequence might be able to sequester, it might be able to sequester some of the RPPGFs that are floating around in solution. So we'll step back. So RPPGFs, we know, can interact with this. And, well, we know it interacts with that, and it interacts with thrombin, but thrombin also interacts with that, trying to cleave it, okay? Just keep that in mind. But this is able to inhibit that cleavage from happening by binding to this. So this is something that this can do. So imagine with me for a little bit. You have these floating around in your body, and they are actually preventing your blood from clotting to a degree. It's something that is keeping your blood less clotable. Now, it's a regulated process, right? But it is one of the things that it's doing is to prevent blood clots. So if you have your spike protein with this sequence on it, it's possible that this sequence is going to be interacting with that peptide that is floating around and sequestering it, basically keeping that peptide tied to this. So this is LQPR. Let's LQPR. So it's right there. Okay. So now keep in mind this is if it's exposed on the spike protein, right? So a lot of parts of the amino or of the protein are not actually exposed to the environment. Sometimes they're nestled inside of the protein itself. And this is especially relevant with the spike protein, which is a trimer, so it covers up a lot of its own sequences. But what could also happen is during proteolytic cleavage, as yeah, as the spikes get cleaved and are degraded, you might have some of these floating around, or this is probably floating around exposed in the environment. And since it looks very similar, so it has 6 out of the 9 residues identical, and it has 8 out of the 9 residues, at least, with a similar chemical property. Since that works, perhaps it will be able to sequester some of the RPPGFs. And if it sequesters some of the RPPGFs, then the thrombin is going to be a little bit more active in the body. Now, that could theoretically make you more prone to clotting, right? If you're getting rid of these in the system that are thinning your blood, sequestering them by the amino acid sequence peptide, that could start clotting your blood a little bit more. So something to look at, right? This is something that someone could get a clue from bioinformatics and then generate a hypothesis and then run an experiment later to see, okay, yes, this works, or no, it does not work. So yeah, I think that is enough of this. I'm going to start this, and I'm going to look at that later. So continuing down, and I will do this for, let's say, another 20-30 minutes. I like this because I'm going to do this anyway, so I might as well record it. So structure-based design of an indolysidin peptide analog with increased protease stability. Let's look at what indolysidin is. So it's a ketonic antimicrobial peptide associated with bovine neutrophils. It consists of only 13 amino acids and has the highest tryptophan content of any known protein. It is amidated at the carboxy terminus in nature. So it's an antimicrobial peptide that is floating around. And so that makes sense with this alignment, right? So you have a bunch of lysines and tryptophans and prolines and another tryptophan, and then you have a tyrosine instead of a tryptophan, but tyrosine and tryptophan have very similar properties. So I'm surprised they don't have that as a plus sign here. And then you have isoleucine and proline, and then another tryptophan. Yeah, like to me, that is very similar. That's a very similar peptide sequence that might have some cross-reactivity. So that's an antimicrobial that is produced by the bovine neurons. So structure-based design of indolysidin. Oh, and that's why it's called indolysidin. So tryptophan has the side group, the side chain is an indole group of tryptophan. And so indolysidin means that it is an antibacterial with a lot of indoles on it. So that makes sense. With increased protease stability, So they're saying is, can we make something that looks like indolysine that is even more powerful than indolysine or more stable than indolysine? So what I would presume is that um, a bunch of bacteria are probably going to be producing proteases that are very effective at breaking this down so that it doesn't kill it. But if you can make peptide analogs, or if you can make different versions with different peptide or different amino acids that are even stronger against proteases, they would do an even better job at killing the bacterium. 2003, like there's a lot that you can learn from research that was done decades ago. So indolycidin is an antimicrobial cationic peptide with a broad spectrum activity isolated from bovine neutrophils. An indolycidin analog, CpDDD, with improved activity against gram-negative bacteria, had increased positive charge and amphipathicity while maintaining the short length of the parent molecule. So here, so they said cationic, which means that it's positively charged. So here they said they made a different analog that has even more positive charge and has even more amphipathicity. So it has both uh, positive charge and it is both hydrophobic and uh, hydrophilic at the same time. And so since it's amphipathic, a lot of times that helps the peptide get in through cellular membranes. So cell membranes, they're like a little fatty membrane with polar groups on the outsides. And this is a way for the cell to remain separated from its outside. But if you have an amphipathic molecule, a lot of those are able to actually get in through the cell membrane without any problem. The structure of CP11, and that is that the name that they gave? Okay, the structure of CP11 in the presence of dodecyl phosphocholine micelles was determined using nuclear magnetic resonance spectroscopy. CP11 was found to be an amphipathic molecule with a U-shaped backbone, bringing the N and C termini in close proximity. So this molecule curves around. Okay, yeah, so here, CP11. So that's what they're talking about. So CP11 is this molecule, and this molecule actually curves around, and that probably helps it get into the cell of some form. And they tested the structure of this in the presence of dodecyl phosphocholine micelles was determined using nuclear magnetic resonance spectroscopy. And yeah, so basically they did an NMR of this inside little bubbles of DCP. So this is a micelle is like a bubble with I think if you're talking about like micellar water, so micelles are basically little fake cells, little fake bubbles, and they use the different types of amphipathic molecules to create those bubbles. Continuing, was found to be amphipathic molecule with a U-shaped backbone and close proximity and a cyclic disulfide bond. So I see. So this cysteine and this cysteine formed the disulfide bond to help make it a circle and was designed to stabilize the lipid-bound structure. Okay, so knowing all this, where does this revealed that the higher trypsin resistance to cyclo-CP11 may be due to more compact packing of the lysine and tryptophan side chains. These findings suggest that cyclization may serve as an important strategy in the rational design of antimicrobial peptides. So discovering new antimicrobial or what would you call it, antibacterials. That is an important research field. And so they're saying here that cyclization of antimicrobial peptides might be a useful strategy. at creating more resistant drugs, for lack of a better word. So I would say that this means pretty much nothing to us because so this is probably chance. I mean, maybe. I mean, could you see a virus utilizing a peptide that kills bacteria? I mean, maybe. So if it does get cleaved and it ends up killing nearby bacteria, I mean, I guess you could make an argument that the bacteria are um, fighting for the same resources, right? So if you kill the neighboring bacteria, maybe the virus is able to get more of the nutrients that are floating around in the serum. I mean, that's a stretch, so I wouldn't go that far, but I mean, possible. Continuing down this. So crystallographic structures of thrombin, complex of thrombin receptor peptides, existence of expected and novel binding modes. So, okay, so this is a different peptide that, so we talked earlier about how this, I think it was PAR1, that receptor had this amino acid sequence that bound to thrombin, and that this sequence was also able to bind with RPBR or something. It was a short peptide like that. So this is basically going through a similar thing. They're analyzing the structure of thrombin complexed with this peptide. So that could be a strategy. It has nothing to do with SARS coronavirus or SARS-CoV-2, but if a lot of times we learn a lot about a protein or enzyme if we can figure out its three-dimensional structure. And one of the ways that we find the three-dimensional structure is that we crystallize the protein and then we shoot it with an X-ray. And then when we shoot that crystal with the X-ray, then the X-rays will sparkle or scatter, and then you collect that data of the scattering, and you can figure out. It's quite amazing. I still have no idea how it works. I learned it at one point in time, and I didn't really learn it. You can, with all those dots that come off of it, you can figure out the 3D structure. But a lot of proteins are very difficult at crystallizing. And so one technique that you can use for analyzing crystal structures or analyzing proteins, is that you can use a peptide, or you can use another protein, or use a chemical that you know binds very strongly with the protein that you want to study. And if you bind it, that can often stabilize that protein so that it's not as squishy or moving around as much. And after you do that, then it becomes easier sometimes to make a crystal out of that protein after you've concentrated it enough. So yeah, that has nothing to do with SARS-CoV-2, but that is just a little bit of knowledge that you're never going to use in your life. But just so you know, that's what some people can do. And here, in this case, they complexed it with the thrombin receptor peptide. Granted, they might have known the structure of thrombin already, but it's one way that you can also learn information on the structure of the protein when it is actually bound to the thing that it likes to bind to. So that can tell you if there's a different structure, unbound and bound. So let's continue. An hour 30. Okay. So, okay. Characterization of P2' and P3' specificities of thrombin using fluorescence-quenched substrates and mapping of subsites by mutagenesis. So, let's look at this. The importance of substrate residues P2' and P3' on thrombus catalysis has been investigated by comparing the hydrolysis of a series of fluorescence quenched substrates. Each consisted of a 10 residue peptide carrying this, which is fluorescent, and this, or maybe this is fluorescent and this is the absorber. I don't know. Cleavage of such a peptide relieves the intramolecularly quenched fluorescence, allowing. This is just a study of molecular, the rates of thrombin. So, yeah, I don't know why. Let's look up PRS. Oh, I think this came up earlier. Oh, shoot. Oh, no. Lost. Find PRF. PRSF. So this, yeah, so it looks like we have gone over this PRSF sequence multiple times before. So we're going to skip that. So this looks new. Interactor with the bovine serum albumin conjugate. Oh, no, I'm not going to, no, that I don't like that website. Conotoxin, so here again, we're going through the cysteine-rich sequence of the end of the spike protein, and it has a lot of similarities to these conotoxins, which also are similar to hepcidin, but not much to be gained from that. And you know what? I think bombalodin. So I believe a peptide that bees can produce. So different bees will produce different types of bombalotins, if I remember correctly. And this can result in, well, I would say if you're allergic to bees, then I would try to avoid getting coronavirus because it has a sequence on the spike protein that is pretty similar to bombalotin 2 specifically. So if you are allergic to this particular amino acid sequence, then there might be a chance that you're going to be allergic to this amino acid sequence as well, because it has, well, it's 53% identical and 73% similar. So it also has three additional amino acids that have at least similar size and or chemical properties. So, and remember, antibodies aren't perfect, right? So if it's similar enough, then it might cross-react. So, and that if you go into anaphylactic shock from bombalotin 2, or I'm gonna search that just to double check. Yeah, American. Yeah, so they're made from bumblebees, and I don't believe bumblebees bite that much. But bombalotin peptides are found in the venom of bumblebees, so there's probably a lot of variety within them. Just be warned, if you're allergic to bees, I would take extra precautions. Um, Continuing, and granted, this piece might not even be exposed on the SARS coronavirus spike. Granted, it is in the S2 subunit, so it could be already cleaved and floating around in solution, especially if it's been broken up by peptides to release, or broken up by proteases to release all those peptides. So, what is, oh, Iraq. I think oh no, that's a different one, but analyzing and simulation of a neutralizing epitope of transmissible gastroenteritis virus. So this is just saying that that amino acid sequence is a part of a different virus that is used to make antibodies. But it probably doesn't mean much. Bombelotin 3, so yeah, another bumblebee or another bee toxin. So if you're allergic to those bee toxins, I would take extra precaution to try to not get that sequence in your body, because that sequence is pretty darn close. In fact, this one's even closer than the previous bombelotin. Oh, but this one's only 13. Yeah, 13 residues, and that's 17. So continuing, interactor with this, however, on alpha beta 2. Yeah, I don't see anything important here. And conotoxin interacts with these monoclonal antibodies, which you probably would buy those. So it looks like we have gotten to the part of the search engine where we're basically looking at more things that are completely unrelated. But I guess not completely. Catechol 1,2-dioxygenase. Maybe that would be something to consider. What about this? This might be. So as you go through these things, you could could be digging nowhere. That's one of the nice, or the things about bioinformatics, is you might not actually be finding anything, right? You could just be going through a bunch of crap, because biology is a very messy and a very noisy study. But so are antibodies, right? So if you're producing antibodies against noisy things, then they might easily cross-react. So reduced tendency to form a beta turn in peptides correlate. Yeah, I'm not interested in that at all. So I think, at least for this one I found interesting, the fibrinogen alpha chain. So fibrinogen is the precursor to forming blood clots. So here we have a sequence that is pretty similar to a portion of the fibrinogen alpha chain. Now, granted, fibrinogen is a very large molecule, if I remember correctly, so it's easy, I guess relatively easy, to have sequences that would partially align. But just be warned, if you are making antibodies against this sequence of the virus, those same antibodies might theoretically be attacking your fibrinogen. Now, would that increase the likelihood of forming blood clots? Would that decrease the likelihood of forming blood clots? Really, that is a very, I wouldn't know where to answer that. But knowing that that sequence is in there with 81% positive rate for that sequence, if you do have an antibody produced against that, it's very likely it could cross-react with fibrinogen that is floating around in your blood. And if antibodies are interacting with fibrinogen molecules, then it could start clumping those fibrinogen molecules together. Granted, it wouldn't. So fibrinogen usually has to be cleaved by thrombin for it to start the polymerization process. But here, you might be able to just bring the fibrinogen together, and maybe it's not an actual clot, but maybe it's something similar. Or if you're bringing them together, it might be easier for thrombin to attack all of them all at the same time and then very quickly form a clot, where normally it would take a lot more time to do that. I don't know the answer there. I just want to, that's something that you could also look into. So the fibrinogen alpha chain has a sequence that is somewhat similar to the SARS coronavirus sequence. And what I could also do is I could search this into this. Oops. So here, and it's also conserved to a large degree. Now, granted, there is a difference between SARS coronavirus and the other coronavirus spikes, in that it has a tyrosine instead of a histidine, but that's a pretty similar chemical. What is it normally? So it's normally QDGKSS. So QDGASS. So what it's looking like here is that through the other coronaviruses, they went from an E to a D, which would make it better at aligning with this. And then let's see what else. DGK. So that lines up well. And remember, we're focusing here. And then alanine instead of serine. So alanine, okay. And then H. Well, yeah, that doesn't mean much. But H would be much closer to, no, not really. Phenylalanine, HF, okay. And then PREG, but it's QKEG. Yeah, so looks like there was one change that made it a little bit closer to fibrinogen, but not that much closer. So you could say that if it happens to the SARS, if there is a result of the SARS-CoV-2 for the reacting with fibrinogen or influencing fibrinogen in one way, shape, or form, and it's done by this sequence, then it would also be done by the other sequences as well. Granted, so keep in mind, we're finding there's a lot of homology between the different coronaviruses, but if you get highly infected by one of these coronaviruses, or one of the more serious coronaviruses like SARS or MERS or SARS-CoV-2, you're getting a lot of the virus, and a lot of it is throughout the entire body, or it's just a very large viral load. So whatever response you make from that is going to be far greater than the response you make from a lot of these other coronaviruses. So that would heighten any effect that you would see in these lesser coronaviruses, long-term effects especially, would also be heightened by the fact that you have had a much more serious disease from the SARS-CoV-2, and you have, especially if a lot of it deals with the immune response, and a lot of the negative side effects are a result of abusing the long-term immune response, then the stronger your immune reaction is, the more serious your long-term complications might be. So, gosh, this is addicting. Maybe just a few more. The VY. The defense function of plant inhibitors are not restricted to insect enzyme inhibition. So 3-plant proteinase. Okay, I think I remember reading this. So basically, these synthetic peptides were able to block some protease activity, but yeah, I don't think that's that important. So I think we've already exhausted the ability of this pepBank to give us more hits. Now, things to take note of: we got quite a few hits on thrombin. Okay, so thrombin, the enzyme that helps create clots. We got a few hits on thrombin and some of the things that thrombin reacts on. We got hits on fibrinogen, so another blood clotting related thing. We got hits on hepcidin, which is something to deal with iron metabolism. And then I really have a hard time remembering all the things that we have gone over. Yeah, so those are the main ones that I can think of right now, because I mean it's a 2-hour session. So later, what I should do is I will go over this sequence right here, where we have extremely good um, homology. Now, that good homology could come from a specific uh, type of secondary or tertiary structure that this particular sequence is very good at doing, which is why it is in two completely unrelated proteins. Uh, Granted, it was not in any of the other closely related coronaviruses, right? Right. So if we go to so LVSSQ, if you go here and you look down, not there's hardly any homology going on between it. There's some, so if you look here, this area is slightly conserved. And if you go in this area is slightly conserved. And then if you look at the other, at least some of the others, this area can be, well, yeah, slightly conserved, not that much. But yeah, this is completely novel. This area is completely novel. Uh, Compared to the other coronaviruses nearby. Now, I can do a blast on this. So what I'm going to do is I will analyze this sequence a little bit more to see if there's anything that can be gained from, if there's any other sequences that are like it. But yeah, to see that spike coronavirus have a series of, what was that, 13 peptides? So say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Oh, shoot. 1 second. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So to have 9 out of 15 be exactly the same, and then to have 1, 2, 3, 4 be like closely related. Yeah, that comes across to me as something that really should be paid attention to, especially since this is a released peptide that just floats around in the body, this NPP peptide. I'm going to ask around, see if this means anything. So, I'm glad I did this because now I can X out a bunch of tabs that are on my computer. This has given me a few different directions and paths that I can do some more research into. Yeah, but what it sounds like to me is that we do know that the ACTH hormone is effectively sequestered when you are infected with some coronaviruses and some influenzas, and that sequestering is done by antibodies. So we know that for a fact. And the rationale that a lot of researchers have said was, okay, look at the RDRPs and look at the SARS spike protein, and you can see you have a bunch of sequences where you share 3 amino acids with the antigenically important part of the ACTH peptide. And like, to me, that doesn't 3 amino acids on the antigenically important part. Like, to me, that really doesn't 3 out of 6. That doesn't mean much to me. But granted, that could be enough to get some cross-reactivity of antibodies. Well, if we're so focused on having only 3 of the 6 energetically important amino acids play a large role in the autoimmune response, then we should look at places where we have 9 an overlap of 9 amino acids, and effectively we have an overlap of about 12 amino acids. I mean, if I look at this and I'd say if I was an antibody, I would, if I can attack one, I'm definitely going to attack the other. So yeah, I will leave that as that. So I mean, perhaps maybe this area is protected, but in some way, shape, or form. I guess we can look on the. I said I was gonna quit, but I'm gonna keep on going. So yeah, with the spike protein, this is the topological domain, and it's all from 13 to 1213. It's all extracellular. So going back, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So from here on out, it is outside of the cell. So from here on out, it's outside of the cell. Now, granted, it could be nestled within the spike protein trimer still. But this is when the spike protein trimer is whole, right? So if the virus has already gotten into the cell, or if the products are being broken apart, so if these spike proteins are floating around and the body is trying to digest them and remove the proteases, well then it's very likely that you're going to end up with a lot of this peptide just floating around in solution. Yeah, so yeah, I'd say this is important, and I believe this sequence was important too. I think I did a search on that, and I might be able to do that later. And you know, this sequence might also be important, and like there's not a lot here. Over here though, it does get a little bit more important. And then over here, this is definitely something that should be considered. So what I'm gonna do is continue my research and drink a lot of wine. I'm gonna get up. I'm gonna enjoy the rest of my day. But thanks for watching. I'm going, and if you have any questions, or if you have any concerns, comments on what I was just doing, any corrections, definitely, because granted, I'm doing this off the cuff right now, and so sometimes I misread, sometimes I don't explain what exactly is going on inside my head when I read this stuff. But yeah, I just thought if I'm going to be doing this research anyway, I might as well record it and provide it for everyone else to critique. So with that, I'm finally able to X out these 30 tabs because I've been holding on to them to do this video. So thanks for watching, and I'll see you later.